Hi, I'm Chris Vulpe. I'm from the Superfund research program at the University of California, Berkeley. And I'm going to tell you a story about some of our work, ongoing work, as part of the Superfund project. And before I start, I'd like to first introduce the people who've really done this work. And this is a collaboration between my group and Luping Zhang's group, who together we have been working on this project to take advantage of some of the unique characteristics and tools that are available for yeast to help us understand the role that genetics play in the susceptibility to toxicants. Most of the work that I'm going to talk about today is the work of a very talented postdoc in my laboratory, Matt North, and he built on the work of William Joe, who established this assay in our lab, and as well as part of a collaboration with Zhufen Ren, who worked in Luping's lab. Environmental exposure to a variety of toxicants can lead to adverse health consequences in people, and this can be modulated both by the age of the individual and the time which they're exposed to that environmental agent. Here in the Superfund research program, we're focusing on the effects of a variety of different toxicants, including organic compounds and metals and metabolites. Now, these are compounds that are frequently found at Superfund sites, and our goal is to better understand the effects of these compounds on people. Now, as I mentioned, certainly the age and time can have a big effect, but one thing that hasn't been looked at as much is the role of individual susceptibility in the response to toxicants. We've been focusing on ways to try to understand how individual differences can affect the response to Superfund toxicants. I'm going to focus in this talk on benzene as an example of this work. So it's clear that everyone is different in our response to toxicants, and this is not surprising given the large genetic differences between people. We think that the individual susceptibility can be affected by genetic factors, and these factors are present in genes and could end up making some individuals more or less susceptible to a toxicant. So in this sort of simple example below, I'm showing a hypothetical toxicant exposure. Now, it's clear that some people, when are exposed to a toxicant, develop disease, and some, even though they're exposed to the exact same exposure, don't develop the disease. This has led to the sort of hypothesis that it may be that these individuals are more susceptible, as shown on the top, and that there may be individuals who are less susceptible that don't result in disease. So why is this important? Well, one of the reasons that this would be important is that it would help us try to identify what are the genes that are important, and perhaps we could look at those individual genes and help predict whether an individual is likely to develop disease or not in response to a toxicant. So where do we look? Well, that's the problem. There are a lot of genes. This is probably an outdated number, but there are about at least 37,000 genes. And how do we figure out which genes are the ones that are important for susceptibility to a particular environmental toxicant? Well, one approach is to take what we already know about the metabolism of a particular toxicant. For example, for many toxicants, we know something about their absorption, their metabolism, such as phase 1 or phase 2 metabolism, and ultimately excretion. And clearly, differences in how an individual does any of these processes could lead to differences in their susceptibility. For example, an individual who did not absorb a toxicant potentially would be less susceptible to that toxicant. However, unfortunately, although we do know something about metabolism, for the vast majority of toxicants, we actually have very little information on how they actually cause their toxic effects, or what's called the mechanisms of toxicity. So what do we do? Our goal is to try to identify the genes which could contribute to individual susceptibility to an environmental toxicant. How do we identify those genes? Here at the Superfund research program, we're taking a novel approach, which is taking advantage of a model organism, yeast, and using that as a way to help identify the genes which could lead to susceptibility to toxicants. So the question is, why would you use yeast? Yeast, or super yeast as we like to call it, shows a conservation of many of the fundamental cellular pathways that are involved in response to toxicants. They are susceptible to toxicants, and perhaps most importantly, they're very easy to use and abuse with various different toxicants. Yeast have been very successfully used for a wide variety of different types of biology and have contributed greatly to our understanding of cell biology, cancer, as well as a whole wide variety of other biologic processes. For whatever reason, they haven't been used as much in toxicology, and we've decided to take advantage of this unique model to help us understand susceptibility to toxicants. So how do we do this? Well, we take advantage of a unique resource for yeast, which is that in yeast, every gene that is present in yeast has been knocked out. So there are approximately 6,500 genes in yeast. Each of these genes has been removed individually and made a strain of yeast which is missing just that gene. So as you can see there with the blue flag, one gene has been removed. Not only has the gene been removed, but it has been uniquely flagged so that you can identify that yeast, that mutant yeast, in a population of all the other mutant yeast. So every gene has been individually knocked out. Every gene has been flagged so that you now have a collection of strains which uniquely identify every mutant. So how do you use this collection of yeast? Well, what you can do is you can grow all of these yeast together in a pool and expose them to a toxicant. And then you can basically ask, well, which of these yeast strains grow well, which ones grow poorly, and which ones don't really care whether that toxicant is present. What this does is it gives us a readout of which genes, which strains, and then subsequently which genes are susceptible to the toxicant. How does this help us? Well, what this allows us to do is it allows us to identify all of the genes that are important for growth or survival in that toxicant. We've now used this approach successfully, which we call parallel deletion analysis, or PDA, to identify the genes which are important for yeast to grow in a whole wide variety of toxicants. And furthermore, we found that for many of these genes, there are human counterparts, or orthologs, which carry out similar functional roles in other organisms, such as mammals. I'm going to talk about one of those toxicants, which we've worked on most recently, which is the benzene. So benzene is a well-known toxicant. It's one of the most highly produced chemicals, and it's found at many of the EPA Superfund sites. It's been known to cause a large number of diseases in people, primarily cancers, and after long-term exposure, whereas high-level exposure actually can be fatal. However, despite long study of this compound, we still are relatively unclear on how benzene causes its toxicity. So we decided to use yeast to help us try to understand the mechanisms of toxicity and ultimately use this information to be able to go back to human populations and be able to test for the presence of variations in particular genes that could modulate toxicity to benzene. So this is a simplified model of benzene metabolism in people. And what's important to note is that there are a series of metabolites which are produced after the benzene is taken up into the body and metabolized in various tissues, including the liver. Previous studies have suggested that these metabolites are probably the metabolites that are responsible at least for a significant portion of the toxicity of benzene. As yeast does not have its own inherent metabolism, or doesn't have metabolism similar to humans, we decided to use these three metabolites on yeast to try to identify the genes which are responsible for susceptibility to these metabolites. The three metabolites that we tested are hydroquinone, catechol, and 1,2,4-benzotriol, which I probably mispronounced. So how do we do these experiments? Well, it's relatively simple. We figure out how much kills these yeast, and we determine what we call an inhibitory concentration, and we treat the yeast at a concentration that's enough to inhibit their growth, but not so much that it causes outright death. We then grow them, expose these pools of yeast where every gene has been knocked out individually, so we have a pool of multiple different strains. We grow them together for a certain period of time, and the time period that we use are generations of growth. So generations just mean the time it takes for the yeast to multiply and produce the next generation. So we expose them for a limited period of time, 5 generations, or a longer period of time, 15 generations. And then we ask, well, what's left? What yeast have survived this abuse? Which ones are resistant, which ones are indifferent, and which ones are sensitive? So what we're looking for is the loss of particular strains in this exposure situation. If they are lost, that means they are less able to grow in that condition, and suggests that the gene that strain is missing is important in the ability of that yeast to tolerate or deal with this toxicant. We define what we call a fitness score, which is just a way of measuring how well the strain can grow. And we define this as being how well it grows in the treatment minus how well it grows in the control. So this is just an example of the data, showing all of the yeast strains here on one side. And then you see the fitness scores along the other side. And you can see that many of these yeast grow quite poorly in the presence of the different metabolites. Now this is showing the results for one of those metabolites, hydroquinone. And again, I'll remind you that the fitness scores here show the ability of the yeast to grow in the presence of the toxicant compared to the ability of the same yeast to grow in the absence of the toxicant. And so this is showing that, for instance, if you have a fitness score of -1, it means that the yeast is growing about 2 times better than it did in the absence of exposure. This is a summary of all our results showing the effects of benzene on the yeast, and it identifies, it's showing all of the genes that are important for the tolerance to the benzene metabolite hydroquinone. So you can see that there are many different components in the yeast which are important for tolerance to benzene. And I'm going to focus now on one of those components which we found, which is called IRA2. And if you remember, that was the one which was at the top of the list. This was the most sensitive strain. Now it turns out that IRA2 is a gene which actually regulates another gene, or the gene product regulates another gene product called RAS. So, of course, when biologists hear the word RAS and hear cancer in the same sentence as we have heard with benzene, we tend to get somewhat excited. So RAS is a very well-known protein, and it's been shown that the deletion of IRA2 leads to increased RAS signaling. Now, this can actually then ultimately result in yeast, as well as other species, in increased cell division. And if you have unregulated RAS, that leads to potentially unregulated cell growth, which is a characteristic of cancer. Shown here on the top is just a confirmation showing that, in fact, IRA2 mutant is very sensitive to hydroquinone as compared to the wild-type strain. So this was very interesting to us, suggesting that maybe there could be some interaction between hydroquinone toxicity and RAS signaling. So we went ahead and tried to verify this. Now, as I mentioned, IRA2 is a mutant which leads to increased RAS signaling, and that's shown here on one side of the screen, A. Now, we showed now that if you overexpress RAS and actually lead to a downregulation of RAS, you actually get increased sensitivity. So together, these results suggest that RAS signaling is important in modulating toxicity. So why is this important? Well, as I mentioned, RAS1 regulates the progression of cell cycle. It suggests that, as we saw before, that hydroquinone has multiple cellular targets. Now, why would RAS signaling lead to increased toxicity? Now, these are just our hypotheses. One possibility is that the hyperactive RAS actually leads to a very rapid cell cycle, which leads to less time for the cell, whether that be a yeast cell or a cancer or a normal cell in a mammalian thing, to repair that damage, which could result in toxicity and ultimately growth inhibition. Conversely, a less active RAS could actually give you time now to correct those errors. So why is this important? Well, this is important in people because a homologue of IRA2 has been identified in people, and this gene is called neurofibromatosis, or NF1, and this is mutant in a disease called neurofibromatosis 1. Now, this is a very common autosomal dominant disorder, which leads to a variety of problems, including predilection to certain malignancies. What struck us was that the type of leukemia that they get, acute myeloid leukemia, which they have an increased predilection to have that, is the same type of cancer which is seen in benzene exposure. And so this led us to wonder whether it's possible that there could be some interaction between NF1 and hydroquinone toxicity. So a test in a mouse model, and these are mice which are, and this was done in collaboration with Kevin Shannon's group at UCSF, these mice have a specific knockout of NF1, but only in the hematopoietic cells. And what we found is that, in fact, these mice, or the cells from these mice, are more susceptible to genetic damage as measured by a measure of DNA damage called the micronuclei assay than a wild-type mouse. So this provides a confirmation that the RAS signaling appears to be important in benzene toxicity, and found an unexpected link between this phenomenon and neurofibromatosis 2. What do we conclude from this study? Well, one, we've identified a lot of the potential modes of action for the various metabolites. As I shown but didn't mention, it appears that oxidative stress is a very important in this process. And as I've mentioned, that RAS signaling modulates the sensitivity to hydroquinone. Furthermore, we showed that the NF1 hematopoietic cells are sensitive to hydroquinone, which suggests that there's a remarkable conservation of fundamental cellular processes going from the yeast all the way to the mammalian cells, and provides a good example of the power of using a model system to help us understand susceptibility to an environmental toxicant. So what are the next steps? Well, hopefully we're going to try to now use this information to go into human populations who have been exposed to benzene. And that will be hopefully the subject of a future presentation. I'd like to thank all of the people who have helped with this work, the Superfund group, my group at UC Berkeley, and the biostatistics group here at UC Berkeley, as well as UC San Francisco, our collaborator Kevin Shannon. Thank you.